As mentioned in the last section, one of the many types of secondary metabolites produced include the endogenous plant hormones. Hormones are what plants use to control the various phases of their growth cycle. So, for example, hormones are involved in controlling the process of flowering and the process of fruit production and seed maturation and germination and leaf production and so on and so forth. And so these are often regulated according to the time of year, according to climate conditions or temperatures and the amount of sunlight in a given day. And so this allows plants to time the various phases of their growth cycle to the most advantageous conditions or times of the year. And these hormone systems work very efficiently because they are produced in small amounts, which means that cells don't have to expend a lot of energy to produce these hormones. And these hormones basically work in that, as it says in your notes, they bind to specific receptors in a cell membrane and they trigger a series of events and usually work by turning genes on or off. So let's say turn genes on or off. And we'll elaborate more on this here in a minute. What are the kind of plant responses that are controlled by hormones? Virtually all plant responses. And so let's make a short list here. These are all controlled by hormones. These responses include things called tropisms, and these are either positive or negative growth in reference to some sort of stimulus. So we'll say growth to or away from a stimulus. And the stimulus could be things like light. In other words, a plant might lean and grow its leaves more towards the light. It might just turn those leaves to get a better angle on capturing some of that light. And that is called phototropism. And another example would be gravity, and otherwise known as gravitropic responses, in which, for example, a seed that gets planted, the roots grow down and the shoot grows upward, and plants stimulate that growth and control that directional growth by using these endogenous hormones. There are a couple other tropisms out there, an interesting one called thigmotropism. That is growth towards or away from some mechanical stimulation. For example, many vining or climbing type plants grow what are called tendrils, and we talked about tendrils early on in this class. And these tendrils grow relatively straight. They might wiggle back and forth a little bit, but when they come into contact with some solid object, you know, a piece of chain link fence or whatever the case may be, this physical contact here, a thigmotropic response would be that this tendril then starts to curl in the proper direction so that it can actually latch onto that object, and that's what's referred to as thigmotropism. And so any kind of tropic response is controlled by hormones. Likewise, other responses that are more commonly thought of, flowering, vegetative growth. After flowering, we have, seed maturation, we'll say seed production. And we could also add fruit production or fruiting. Shoot growth is tightly controlled, and we'll talk a specific example of that in a few minutes. Also, a couple of what we would consider to be maybe more negative type growth responses, and that would include dormancy. And dormancy is basically just shutting down in parts of the year, most typically that it is not so hospitable for the growth of the plant, i.e., trees dropping their leaves prior to the winter months. And while we're on the topic of dropping leaves, that is referred to as senescence. Okay, and you'll want to write that down, and that just basically is programmed death of various plant parts, and it just means a gradual breakdown of anything that the plant can recycle out of those leaves, for example, and that's what causes the color changes in the fall on a lot of deciduous plants. The chlorophyll gets broken down, and so what remains is mostly the xanthophylls and carotenoids, which are red and orange and yellow in color, and that's what causes the leaves to change in the fall. One final one is what follows senescence, and that is abscission. And abscission is basically this sounds a little morbid, but it's pretty much for plants self-amputation of the senescent parts of the plant. So in other words, a leaf starts to senesce, the plant starts to break down anything it can recycle out of that leaf. And then we have what's called leaf abscission, which means the tree drops the leaf. So leaf abscission is dropping of the leaves. Fruit abscission is when the fruit ripens enough to where the plant drops the fruit from itself. And so if you're ever asked to, hint, hint, give me a list of plant growth responses that are controlled by hormones, you could pretty much just figure out any kind of plant response at all and write it down, because they are virtually all controlled by these hormones. And one final thing to note is this term endogenous, and this just means that all plants produce these particular hormones. They're found in all plants. Now, how do these hormone signals work? Well, they're just chemicals and mostly organic type molecules, and the plants produce them on an as needed basis. Well, the whole mechanism works based on two key factors. We have the concentration factor, and concentration just simply means there has to be a change from some baseline concentration. So we'll say a change, I'll write exactly that, from a baseline concentration. In the case of hormones, most of the time this is a change from a very low concentration to a high concentration. So low or virtually non-existent concentration of a particular hormone, let's say hormone X, to trigger a response potentially in a tissue, there has to be some change. In other words, the tissue has to know that, hey, something is different than it was before. And usually that just means a production of more of that hormone, and so we would say from low to high concentration. This is not referring to movement like we talked about with diffusion, so don't confuse the two. This just means a change from an initial concentration. Concentration must be very precisely controlled because, again, in order to be energy efficient, the plant uses very low concentrations or very slight upticks in concentrations to send messages from one part of the plant to the other. And the other main factor is sensitivity. This has to do with the fact that not all tissues in the plant are going to respond the same to the same change in concentration. And as it says in your notes, the sensitivity of a tissue is greatly dependent on the maturity of the tissue and also the type of tissue. For example, roots respond often differently to a change in concentration of a hormone than would a shoot, for example. Than shoots. To get a little more technical on the subject of sensitivity, what determines sensitivity? Well, sensitivity is determined by the number of receptor sites and also the types of receptors. To talk about both of these together in an example is a television broadcast or a radio broadcast. Some of you are maybe only used to watching television over cable or by satellite dish. Not too long ago, you got your TV basically through the air. just like a radio signal that you probably still use from time to time in your car when you're driving around. And so let's look at it in terms of that. Whether it's a TV or a radio or whatever type signal, you've got a source for that signal. And that signal goes out through the air in the form of waves. Now, that would be, for example, the hormone. Hormone originates from a source, and it sends a signal out to surrounding cells or cells in other parts of the plant. Well, the further away from the signal, okay, and this is probably not the greatest way to depict this, but I'm going to draw these waves as getting smaller and smaller and smaller. In effect, the further away you get from the source of that signal, the weaker the signal gets. Okay, so it's weak over there, it would be strong over here. Okay, so concentration will change based on where a tissue is located and based on where the source of that hormone signal is. But you have to be able to detect that there has been a change in the first place of that signal. And in order to do that, you have to have the right type of receptor. And so, for example, if this were a television signal and I had a TV with an antenna, I could get that signal if I had it tuned in to the right channel. Now, if I had, here's my drawing of an old-timey radio. If I just had a radio, I wouldn't get that signal because it's not made to pick up the TV signals. But the television is, as long as you have the proper antenna hooked up. And so this has to do with sensitivity. Are the right type of receptors in place? So let's say maybe in the roots, we'll say the radio might be an example of the roots, and the TV might be an example of leaf tissue. Leaves, plural. And so there's a case of having the right type of receptor. And so where does the number of receptors come into play? Well, the TV and radio example doesn't play so well into the number of receptors being a factor. And that just allows you to fine-tune and tell the strength of the signal. Remember, these are not waves. These are chemical molecules that are moving around inside of plants. And so let's say we have a cell, and on the surface of that cell, and this is indeed how it actually works, we have receptors. Okay? We can draw some red receptors. I'll draw some of this, some in this pink color, and maybe we've got some in a third type of molecule. Well, let's say that we have a signal of hormone X. Okay. Each one of these little X's represents a molecule of hormone X. So we have to first have some change in a baseline concentration. So if the baseline concentration is 0, these red receptors are not receiving any signal at all from hormone X that they are specifically designed to sense, like a TV is designed to pick up a TV signal. So a change in concentration occurs if suddenly there are more of these X's floating around that were produced by another cell. So if that's the case and one of these receptors is present and it comes into contact with hormone X, well then that can tell us is, hey, there is a change in concentration. Now, in order to detect the magnitude of that change, in other words, to allow a hormone to give more than one kind of signal based on concentration, you have to have multiple receptors to determine the relative strength or weakness. So, for example, if all we had was one of these red receptor molecules on this particular cell, and even one molecule of hormone X came into contact with it, we could basically start some activity within this cell in the form of turning on or turning off the genes of that cell. But that would be limited to just an on-off switch. By having multiple red receptors for hormone X, we can, if say, 3 out of 10 or so are triggered, well, that tells us that there's a change, but also a relative strength. it could possibly trigger as many as 10 if we have 10 receptors, and so it's on a 3 out of 10 in terms of the strength of that signal. And so by having not only determining whether there's a signal at all, but also being able to be sensitive enough through the number of receptors that a cell has, it allows that cell to interpret also additional information about that symbol in terms of where it is on a scale of, say, 1 to 10 or 1 to 100 or whatever you want that scale to be. Now, once it receives its signal, that triggers a series of events and turns the genes on or off.